Hi, my name is Michael Edwards. I was, as Matt says, I have my own bioinformatics consulting firm called Bioinfo Solutions. I used to be assistant professor here at UC Denver. I see some faces that I know. I basically do kind of, and I specialized in bioinformatics. I've worked with a lot of people on campus. I do pretty much the same thing, except this time I don't have to write grants and go to faculty meetings, which is pretty nice. I also get to work in my pajamas if I want, because I work from home, so it's really cool. So today what I want to do is I kind of want to give you the big picture of what's going on with bioinformatics. And I want to use this tool, the correlation engine, to kind of guide you through that. How many bioinformaticians are here today? Yeah, a few. So some of this stuff, this is tailored to those people that actually don't do programming. So just a little background on myself. I started working on genomics or microarrays in 2000 at the University of Wisconsin. I come from the bench. I've killed lots and lots of mice and fed cells and run westerns, and that was my world. About 2000, the microarrays, the ability to measure the gene expression of all genes in the human genome, came out. I kind of embraced that. I didn't have any programming skills or very extensive statistical or mathematical background, but I thought, you know what, I'm going to jump in. This is where the future is heading. That I would rather look at what all the genes are doing rather than just one. And this software allows me to do that. Since I don't program myself, this software allows me to basically analyze this DNA. And what I'm going to do is kind of show you some of the background on, what we're actually, some of the things we're analyzing. I'll give you an outline for this. I also want to make you aware that I have a YouTube channel that I am recording this. This is what this microphone is for. I'm not going to sing, I promise. Unless you want me to. No hands. Okay, never mind. So you can come back and you can actually watch this lecture. Not only can you watch this lecture, but you can watch lots and lots of other lectures on a lot of the other tools that I use. So not only do I work a lot with the base, the Illumina set of tools, but I also work with Ingenuity Pathway Analysis and Partech Genomics Suite. So there's some other tutorials on there. So if I don't answer your questions now, you can go to this YouTube channel and hopefully get the answers. Quickly, just some disclaimers. I always have to say this. The opinions expressed by me are mine and not Illumina's. Also, they reimburse me for these events. I do give talks, but it's just for travel expenses. And obviously the free meals that come along with this. Especially when they go for the corporate, at night, all the corporate guys go out. And so I always tend to tag along and end up on their corporate bills, which is really nice. But no direct pay. So what I'm going to talk about, and I've kind of reworked this talk a little bit, is that what I don't want to do is I don't want to get in the weeds of, the program. I kind of want to at first kind of give you a big picture and basically explain why in the heck would you want to do this in the first place. Why is bioinformatics important? Second, what are the kinds of information do we analyze, right? There's more than DNA. There's a lot more than sequencing. So I'm going to briefly discuss this, and this will be reviews for some people, but for some people, this could be relative, or something they might be interested to know. Also, where's this information stored? Where do we get all this information, right? And how do we access the information? I'm going to, again, go over some of the tools that we use, or at least this tool, to show you how we access all that information, take all that information and come up with like biological, actionable things that we can actually do. And once we have all this information, what do we find? What do we look for when we actually get a huge gene list? And some of these gene lists can be, over 1,000 different genes that are different in a situation. How do you make biological sense of that? And I'm going to tell you right now, that's where I specialize, because I'm a biologist at heart. That's what I want to know. And it always kills me. I see researchers out there, they spend thousands and thousands of dollars in man hours and making postdocs cry, getting this research study together, And at the very end, when they have everything sequenced, they give it to somebody whose specialty is mathematics or statistics, right? Or computers. If you have your research project, you should be looking at your data. You should not be giving this to somebody else. And that's why these tools are developed, is because most PIs don't program, and I don't program. Bless those people that do. Zoo? Oh, he left. There he is. Bless you, Zoo. I appreciate you. And all the other people that program. Hallelujah. Hallelujah. But I don't want to program. I absolutely don't. And sometimes finding that biological insight is a lot different than actually programming. It's a different process, right? One's more, A to B to C. Once you get into the analytical part, it starts turning into spider webs, right? So those processes are different. How can I get to the spider webs without having to go through the programming part, or basically building the tool? So first question I asked, why should I care about bioinformatics? And, I give this talk to, some general audiences too. So, you're all pretty much aware of why we're doing bioinformatics. But I think this slide kind of, very easy way to figure out why we do this. So given up here is the Illumina, it's called Understanding My Genome. This is my genome. Has anybody else had their genome sequenced? Anybody? Cool, I'm special. You haven't even had your genome sequenced. Guess what? You will. And it's going to happen sooner than later, right? There's a lot of information. You can see, here's my genome, and I can go through here and I can actually look and see some of the things that are, some of the traits that I have. One of the traits, every time you log in, it always gives you something new about your genome that it'll tell you about. And it says, based on your DNA, we see you are less likely to have flush reaction if you drink alcohol. I'm Irish. I know this. Right? It's not just that, right? We can get to the little ends, right? And all these companies are coming out, 23andMe, Ancestry DNA, right? There's a tremendous interest in figuring out, knowing what's in your DNA. And when I say that you all probably get your DNA sequenced, is that, when we first sequenced the first human genome, to sequence the first human genome took like 10 years and $10 billion. Zoo, this is always your quote. Is that right? 10 years, $10 billion? 3 billion. 3 billion. Okay. I was off by 7 billion. Now what we can do is we can actually sequence some of the DNA, and it's getting to be about $1,000. And we can probably do this in a couple of days. And as you can see, the hunger to basically utilize this information is not just from like hospitals and the medical department, but it's actually individuals. There's even ones where you can actually, it'll guide, your eating. So I found this when I was searching for some genetic testing sites. Are you still struggling to lose weight? The secret's in your DNA, right? This is coming, and there's a lot of power in this. That, you could look at this and say, oh, they're overshooting, but not really. Genes that metabolize your food, you might have a variation that you might want to eat a certain other type of food or a certain type of other metabolite. And I say this to a lot of people. Some people don't believe me, but I bet this is going to come to the dating sites at some point, right? Match.com. Right? If you look at, eHarmony, 29 dimensions of compatibility. Imagine 6 billion, dimensions of compatibility. This is coming. The public wants this. We, as the medical field, better figure out, how do we analyze this? How do we keep people from hurting themselves, right? If you told somebody, if they had their DNA analyzed and they say, well, you have a 20% chance of, early onset Alzheimer's, if they hear that, you're the general public, you pretty much think you're going to get Alzheimer's, right? So not only do we have to, figure this stuff out, but we also have to teach basically the general population about this. And I think these are, some of these non-programming tools are the way to do it. So, but it's not that easy, right? There's 6 billion nucleotides in your DNA. How do we take all that information and figure out, if say you're potentially going to get a disease, or say if you're vulnerable to a certain type of infection, or you're going to go bald later on in life. You know, the lucky thing for us is that most of this stack of needles is rubbish. I actually got this as a pie chart. It was from it was talking about this. It was a brief history of rubbish, the history of our DNA. So if you actually look at our DNA, most of it actually is useless. Yeah, actually 0.2% of it are active other organisms actually living inside of our DNA, like hitchhikers. There's lots of fossils from previous invasions. We have canine, we have feline DNA in our genome. And when I tell this to, I also teach high schools. I teach bioinformatics to high school, believe it or not. And when I tell the students that, they say, oh, we got cat DNA. Can it get activated and then we can have like super side or something like that? I was like, no, it doesn't work that way. Most of it's garbage. Basically, most of it's garbage. Most of it's repetitive DNA. Only 1% of it actually codes genes, right? And you might look at that and say, well, that's nothing. It's not just the DNA itself, and it's not just the DNA or the sequence of these genes. There's a central dogma of biology, and this is held for many years. It has changed, but again, we're talking about 6.4 billion nucleotides. Again, we can measure all this, which transcribes RNA, the genes. There's about 25,000 genes in the DNA, which transcribes messenger RNA, which we can also measure. This messenger RNA gets translated into proteins, and there's about, just in its purest form, maybe 500,000 human proteins, right? Just this information alone, just with that 1% of the genome, is huge, right? But that's not it. We've discovered that there are microRNAs. So not only do we have this process, but we also have other things that we can measure that also modulate this process. These microRNAs will actually degrade particular species of mRNA depending on what it was engineered for or what it was coded for. RNA, it's not just one RNA, but these pieces of messenger RNA can be spliced and they can be rearranged into many different forms. Right now I'm working on a gene that has 11 different isoforms of it. 11 different copies of its messenger RNA, and they all kind of do something different. So there's complexity not only in just the amount, but even in you just take one unit of that, and that can also be modulated as well. Proteins are a mess, and that's why I try to stay away from proteins. Anybody work on proteins, proteomics? I'm sorry. It's a mess. I did a postdoc in proteomics. Proteins are like cars. You can modulate them any way you want. You can put headers on them. You can put, dual exhaust. You can put all these different things, ubiquitination, hydrolation, phosphorylation. It all does something different to the protein. And again, these are things that we can all measure. Let's go back to the DNA. Not only is it the sequence of the DNA that matters and the actual things that it codes, but it's the way it's packed. That if I took DNA out of one of my cells and I stretched it lengthwise, it'd be taller than me. In order for your DNA to fit in your cell, it has to be packed. And the way it's packed basically determines a lot of its function. And the way you can pack that with methylation, there's a lot of methylation studies out there. We can determine what areas of the DNA are methylated. And this matters. So given here, I hope people can see that. It might be a little too low. These are two insects. One is a locust, and one is just your regular old grasshopper, right? The locust, kind of aggressive, scurries around a lot, flies long distances, forms swarms, can devastate entire communities by eating all their crops. This one, the grasshopper, local, pretty mellow, might fly up in your face as you're riding your bike, bug you, but pretty much very calm insect. When you look at these two, these are, from a genetic standpoint, they're exactly genetically identical. The only difference between these two insects is the way the DNA is packed. And we'll see that a lot with different types of cancer. why I show you this is all of this stuff we can measure, and all of this stuff we have access to through some of these programs that we can use. And not only do we have access to all this information, but in lots and lots of different areas. And one of those places where we deposit all this information is the Gene Expression Omnibus. Everybody's heard of that? Anybody has not heard of it? Good. This is where we're sticking all our data, people. The problem with Gene Expression Omnibus, well, let me basically tell you right here. So this is how many data sets are in the Gene Expression Omnibus right now. There are approximately about 74,000 different experiments containing almost 2 million samples. Has anybody tried to get this information out of Gene Expression Omnibus? Yeah, what do you think? It's not easy. It's horrible. Some of the, formatting can have issues. Some of the clinical information is not in the best place that you would want it. The tools that they use to analyze this on the site aren't very adequate. But this is where it is, and this is where a lot of biotechnology right now is concentrating on. There are lots and lots of cures in this right now. That science could stop tomorrow. We could stop killing mice, growing cells, and I would have a career for the next 2 years. There is a lot of data out here. So basically, during my talk, what I'm going to try to get across to you is how do we utilize all this information? There's millions and millions of dollars within here, and you might think that your research, study is very special, but I'm willing to bet that this can. So the way I kind of think of bioinformatics and all this information is kind of like pixels, right? That each little piece of information really doesn't do much on its own. That if you look at your typical flat screen, high definition television, it probably has a little more than 2 million pixels in it, right? 2 million boxes. When you watch your TV, do you see all those boxes? No, of course not, right? Your head puts that together. It takes all that information, you see pictures. That is what I'm trying to do with all this software, and that's what I do at the back end, is how do we take all of these bits of information and put it together into something that we can understand, that we can use to drive the next potential cure or, maybe a grant proposal. This is my goal, and that's what I try to get to when I actually analyze these data sets. What's a useful piece of information that I can actually inform myself or inform the situation?